So I'm a bioinformatician, and I'm also a product manager. This gives me the opportunity to talk big, not to focus on any of the specifics. We're going to go fast. I've got 15 minutes. I've got my colleagues Chris and Rachel, and they're going to be delighted to jump in and answer any of the questions. So I'm coming from Oxford Nanopore Technologies. At Oxford Nanopore, we perform DNA sequencing. The way we do DNA sequencing, as Adam Philippi introduced yesterday, is we've got a fist. This is your nanopore. We've got a 2-kilometer cable of DNA sequence, and we run it through at hundreds of feet a second. Technology is really cool. It's miniaturized. And, you know, hey, we've got a beautiful marketing slide that's trying to give us the essence of what is the technology and how does it actually work. Okay, technology, marketing, base calling, it just gets, you know, to a point. Let's actually have a look at the individual DNA sequencing device. This is the flow cell. The flow cell is what we generate data on. It's small, it's inexpensive, and it allows us to produce data that comes in a wide variety of different sizes, different shapes. But essentially, what we're imagining is they could be anywhere between sort of perhaps 1 and 50 million sequence reads. There can be a massive diversity in the actual lengths of the sequences. And we need to begin to imagine how can we, as a technology, help our users move in interesting sorts of directions. So one of the first things we had to have a think about is what are the user personas that we need to really address? At the top of the screen here, you can see that our corporate goal is to enable the analysis of anything by anyone, anywhere. We have DNA sequencing devices that we can take out into the field. We've got DNA sequencing devices that have been to the International Space Station. We have people DNA sequencing in hostile environments, being both the Antarctic, the Arctic, being in, you know, all sorts of hostile marine and desert environments. This means that if we're truly going to democratize the sequencing, should we also not be bringing bioinformatics to these same users? This means that the users, shown in the first panel, could range from molecular biologists who are doing field research, through to astronauts who are sort of floating around in the space station and need to understand, is that biofilm that is rapidly proliferating on their spacecraft going to generate a big problem, and should they stay in space forever? Okay, that's perhaps a bit far-fetched, but we have got a number of personas. We've got a number of users that include both the bioinformatics professionals and people who aspire to at least getting some results out of their bioinformatics process. So, okay, perhaps we can address anyone. The next thing is how we're actually going to facilitate this bioinformatics. We've heard a lot about cloud. Yes, send everything into the cloud. But actually, if you're in the desert and your nearest cell phone tower is a long, long way away, then probably cloud isn't the right sort of solution. So this is where we've been trying to challenge some of the dogma and say, okay, bioinformatics, should we not actually do this on the sequencing device? Should we not do this on the computer that is attached to the sequencing device? Yes. The majority of our users are Windows users. Should we be supporting or facilitating bioinformatics on Windows? Heresy, you know, horrible thought. Why would you possibly do that? But hey, 80% of our users are using Windows, and if we're trying to exclude them from the ecosystem, then there's a little bit of a challenge. So we need to support different platforms, we need to support cloud, and then we need to look at the magnificent diversity of what are the sort of science and workflow that we should be supporting, tolerating, or encouraging. So what is our solution to this? Our solution to this is something called Epitome. See, Chris, you know, in the next break, you can see it's shiny, it's beautiful, it's on the desktop. But this is our platform that we use to help all of those user personas get towards a tractable bioinformatics analysis. It is a standalone desktop application. We should have several asterisks here because it's not truly standalone. If you're on Windows, you need WSL. WSL comes crammed full of interesting things like, you know, we've got Docker stuff, and we've got Java, and we've got Nextflow, we've got all these complicated things, but essentially it's a standalone application with a terrific amount of engineering behind it to hopefully provide our users with a one-click experience towards bioinformatics. So, okay, it's a dull slide. Let's move on to things which are a little bit more interesting. So last year, I had the great pleasure of joining some of you folks in Barcelona. And at Barcelona, we outed Oxford Nanopore as a company that was using Nextflow, and we talked about some of the warts in the Nextflow ecosystem and some of the warts in our implementation. But essentially, we had a really ugly solution to helping users move forwards. We had engineered into a Jupyter environment an ability to run Nextflow workflows to produce these reports, and we had this kind of wonky sort of ecosystem that was sort of working. You know, it wasn't too bad. Everything was Docker and Conda, and we've been pulling our hair out. You can see, not much hair left. How do we actually support users in Conda? Conda, you know, it's great, but it's also rubbish at exactly the same time. So, you know, yeah, okay, yep. We had, you know, things sort of working out, and we said the horizon is going to be an integrated application. We're going to move everything over towards Electron. It's going to be beautiful. We're not going to have any of the horrors of Jupyter. Yes. Yeah, we had kind of a lot of fun last time. So this time, I'm just going to give us a little bit of an update as to where we are at the moment. And, you know, are we still happy with Nextflow, or are we going to have to implement our own solution? Well, I'm talking here, so we probably know the answer. Anyhow, Electron app, Epitome desktop, open source. It's friendly, accessible. We launched it at our London Calling event in June this year. It's colorful, it's beautiful. It's designed for non-bioinformaticians. It's designed for people who don't have the sort of resources of the Broad or the Fred Hutch. It's designed for people who, you know, we want to get started with bioinformatics. We want a button to push, but we don't have administrators, we don't have helpers who can help us through the problems. We want to make it easy to get started. And really here, getting started with bioinformatics is trying to help users generate a colorful figure with as few clicks as possible to give them this sense of control. Beautiful figure, I'm in control of my data. I understand that the sequence quality was adequate, I understand the results are fine, we're in a position that we can move forwards and proceed through the ecosystem. Anyhow, majority of folks in this room here are bioinformaticians. You don't want a single button to click. You want to hear a little bit about, actually, how can you use some of this nanopore goodness from the command line, from your own data centers. So we have what we call Epitome workflows. The Epitome workflows are just normal Nextflow workflows. We are somewhat adherent to some of the NF core standards. We do very much like the NF core schema. This provides us with a framework that is somewhat interoperable, but somewhat not. It's designed to be something that makes a lot of sense for our users, and it's really working towards what are the best practices. We want the code to be literate. We want to help you, the professional bioinformaticians, see how we, the nanopore experts, would recommend that you solve particular problems with the particular software environments that have been tested and validated by our own teams. We appreciate that command line is probably the cleanest way to go. With the command line, you can do the orchestration. You can go and integrate with the various systems that you have in-house. So really, Chris and his team are very much thinking about how can we actually run things in our own environment. We have our own HPC. We have an ability to use CPU, GPU nodes. We've got all sorts of different flavors of goodness in-house. And we find that having Nextflow, our sort of epitome flow, workflows, you know, that's the cleanest way to get started. It all makes sense, and it appears to be helping us towards the democratization of bioinformatics. The key point here is that we are not producing a black box, we are not supporting a black box, and everything is going straight through to GitHub. GitHub is where all the goodness lives. 18 workflows, they are supported, and we encourage you to have a play. We want to have merge requests. We want to have pointers as to things that are broken. We really want to sort of understand what we can do to make our products a little bit more appropriate to your own needs. On the left-hand side, we've also got our own web page. The web pages are trying to describe the product. And again, here, we're trying to create a little bit of community amongst these neo-informaticians. These are the biologists who want to do a little bit of bioinformatics, don't really know what anything really means. So we're trying to create blog posts. We're trying to draw the users into the story. We're trying to educate, and we're trying to demystify some of what is going on in the ecosystem. So anyhow, labs.epitome.io, a great place to go. It's going to point you onto the GitHub, and there is all of the goodness. So let's have a look at what are the sort of workflows that we're trying to support. We've got 18 different workflows. I don't have 18 slides because that would get a little bit tedious after about number 2. I just wanted to give a little bit of a sort of insight as to the diversity that we're trying to support. So this slide that looks very different on my desktop to the way it looks here, go figure, it's talking about amplicon sequencing. So amplicon sequencing is just you do a PCR and you want to sequence your PCR product. Really simple, really trivial. The amount of money that people are spending on doing this with other technologies, such as Sanger sequencing, you could actually do this with our technology, a little bit cheaper. We're trying to release a workflow here that takes the canon of an old, tried, tested technology, and we're trying to reinvigorate it for today. So, simple workflow. We've got an integrated protocol that works on the molecular biology side, through to the informatics that you can run on the same sequencing device or the sequencing machine that you have. We're aiming to go and consume the data, to report it, to make it visible, functional, and literate, so that a user can go, yep, this amplicon is exactly what I thought it was. I see no evidence for an inversion. I see no evidence for any nucleotides being lost. I'm happy. Let's proceed with the molecular biology or the rest of my study. So amplicon sequencing, great example of simple science that we're trying to open up to our users who are not experienced in sequence alignment or in de novo assembly of short fragments. At the other end of the spectrum, we have whole human genome sequencing. So whole human genome sequencing has been mentioned a number of times in this forum. The concept is you start off with a blood draw, you start off with a piece of tissue, you want to get towards the genome, and you want to call different genetic variants. We've got a sort of a nanopore marketing campaign at the moment, and it's called, what you're missing matters. Nanopore sequencing gives you ability to look natively at the methylation. You can get SNPs, you can get structural variants, you can get copy number variants. With all of these different sorts of variation, not to forget, of course, variable number tandem repeats or the VNTRs or we've got loads and loads of different genetic variation. So we want to have an integrated workflow where, of course, you start off with your sample, you sequence, the machine goes ping, click on the button, you process the data, and then you have the collection of variant files. You've got the methylation data. You can come up with all sorts of magical, colorful plots. You have the ability to understand what is the etiology of this rare disease, what are the particular functional variants that we should be characterizing or segmenting. So, hey, beautiful piece of work, and it's really making the analysis of whole human genomes a single click for the people who are beginning to get started with their genome analyses. So we've been talking about bioinformatics on the box. We've been talking about bioinformatics, on your sort of laptop. You can do it anywhere. But one of the things that we've been working towards recently is the concept that we need to run all of our workflows in the cloud. So cloud is both great and it's terrible, depending upon where you are and the regulatory environment that you're operating in. I'm often dealing with Windows users. A Windows laptop, when you're trying to process single cell type data, does not for happy users make. We need to give them the ability to have a seamless story where they can go from problem data through to colorful report and the ability to go and open up the Loop browser. How do we do this? Well, we're offering our own cloud sort of plugin. What you do, you're presented with a button. You log in. Would you like to run this analysis on your device, or would you like to run it in the cloud? Hey, I want to run this in the cloud. Machine does a little bit of chewing, and it gives you a complicated number that is an instance or something, or something technical in the cloud. You have to ask Chris exactly what's going on. But from this colorful picture, you then have your workflow running. You get the green bars that individual processes have run to completion. We are in control. Everything is looking great. And the only difference is when we look through some of the log files, we can see that we actually have particular Amazon endpoints where all of the data has been orchestrated and fed forward. This is designed to be a product that is accessible to all of our users, and we have this as an integral part of our Epitome product, and this will be going live in the next few weeks. So what else is coming? Well, hey, we've been talking about Nextflow. We've been talking about how we can begin to sort of integrate Nextflow workflows to make life easier for our users. We're now at the stage where we're beginning to integrate our platform with all sorts of third parties. This brings additional interpretative context to the analysis. Of course, we're upskilling all of these third parties in how to use our Nextflow products, but this is beginning to open up all sorts of new opportunities, challenges and ways that we can collectively move forwards. So super exciting stuff. I mentioned last time that yes, it would be great. We want to help you get your workflows into the Epitome environment. Is this not a way that we can all share our favorite workflows with users who are not quite ready to get started in true bioinformatics? We have a couple of contributions to our epitome environment. The first one is a piece of software called EMU. EMU performs 16S ribosomal RNA classification. Really simple sort of science, but it's a really nice plugin, and it is an alternative to the more traditional Kraken sort of KMA type classifications that have been used. So hey, everything's going in the right direction. I'm 20 seconds already over. Yeah, we've got a load of summary points. Everything is in control. Everything is wonderful. The final ticket here for sort of Paolo was the fact that, yes, we are still meant to be implementing a perfect workflow management software. We're not quite there yet. Chris didn't give me anything mean to say this time. We'll save that for next time. Thank you very much.
Thank you for the very energetic talk and very informative. One quick question, because I'm sure a lot of people are wondering, the epitome functionality that sends the analysis to the cloud, do you pay for it? Do we pay for it? Like, how does the billing work on that?
Yeah, there are certain questions that you really shouldn't ask. The answer at the moment is this is a service to run our workflows at our cost for our customers. This could change at some point in the future. It depends how many people run our workflows. Yeah, I can imagine that. Thank you so much. If there are any more questions, please ask in Slack. Thank you.